welcome to another in our Horizon series of webinars. LRIC started this series to look at the big things affecting livestock sector in Ontario, and we've had tremendous success as far as people attending and tremendous support from speakers. Today is no different. I did a bit of a tallying. It's interesting to me that for our first 7 webinars, considering the number of people that have watched in person or on our website, we are now approaching 900 people that have been affected by our Horizon series. We think that's fantastic. These are the big things that livestock needs to be working together on to ensure a bright future. And so it's good to get this information into the hands of everybody. We have Chloe Neudorf, who will be introducing our speaker today. And if during the webinar you have questions, please put them in the Q&A function. And at the end of the webinar, Jean Howden, who's also with LRIC, will be bringing those questions to our speaker. Chloe, over to you.
Thank you, Mike. Today we are pleased to welcome Dr. Shayan Sharif, Professor and Associate Dean of Research Graduate Studies in the Ontario Veterinary College at the University of Guelph. Dr. Sharif is a contributing editor to the 8th white paper in the Horizon series and is here to speak on that topic, the future of livestock vaccines. Dr. Sharif is a professor of poultry immunology. His research focuses on chicken immune system recognition and response to viral pathogens. As a leader of the University of Guelph's Poultry Health Research Network, Dr. Sharif manages a large network of poultry experts and focuses on research and industry collaboration. Shayan has published over 200 papers and has been working at the University of Guelph for 20 years in many roles. Dr. Shayan Sharif is LRIC's livestock vaccines expert.
All right, thanks very much, Chloe. Much appreciated, and also much appreciate Mike's invitation to be here today. And I'm so glad to be here. Feeling quite privileged to have the opportunity to interact, even though virtually, with the audience, and I see quite a few people attending. So first and foremost, I just wanted to give you a perspective on the future of livestock vaccines. And this is kind of actually culminating from some of the discussions I've had with Mike over the last 2 years or so and his vision for livestock 2030. So we're going to be talking a little bit futuristic, but at the same time, we are students of history. We always need to understand history because history informs our future. So vaccination is not a new topic. It's not a new concept. And as a matter of fact, since 900 AD, the Chinese have been doing vaccinations. And also in the Middle East and also in the Near East, there have been a lot of work done on vaccination since the 900 AD up to the time of Edward Jenner, who in the 18th century did the first modern vaccination. But Louis Pasteur is indeed the person, the scientist, who should be acknowledged and regarded as the father of vaccinology. This picture must have been taken, or at least drawn, in 1885, where one of his associates, and Louis Pasteur is the man with his glasses in the background, and one of his associates is injecting an attenuated vaccine in the skin, under the skin of a patient who had been attacked by a rabid dog. But the fact of the matter is that, first of all, I said it's an attenuated vaccine. At the time, Pasteur really thought that it must be a live attenuated vaccine, not knowing that he was actually dealing with a killed vaccine. As a matter of fact, he didn't even know that rabies was caused by a virus. He had no idea about any of those. But this brave man and this fantastic experimentalist did something that most of us would not even fathom doing. But prior to doing this, you would be astonished to know that Louis Pasteur had actually already developed by this time 2 other vaccines successfully used in veterinary medicine, one of which was against Pasteurella multocida that causes fowl cholera. And the other one is a vaccine against anthrax. And in both cases, he managed to confer significant protection against both of those diseases in avian species, in poultry, and also in mammalian species in the case of anthrax. So in terms of different vaccine types that are currently available, either for human medicine or for veterinary medicine, I would say there are probably 12, 13 different kinds of vaccines that are available, generally speaking. The most important ones in terms of how widely use they are. There are live attenuated vaccines, killed vaccines, toxoids. So the top 3 vaccines that are commonly used for childhood vaccination, and also perhaps I would say many of us or all of us have been vaccinated with all 3 of those, with live attenuated vaccines, killed vaccines, and toxoid vaccines. And up to quite recently, I would have probably said the ones that are identified with a green arrow, subunit vaccines and protein conjugate vaccines, some of us had already been vaccinated with some of these types of vaccines, but not very widespread use of these kinds of vaccines. And up to probably 18 months ago, I would have said it's almost impossible or very unlikely that we would have been vaccinated with a nucleic acid vaccine or with a viral vector vaccine. But because of the unfortunate thing that we are currently focusing on most of our attention, which is the COVID pandemic, many of us have already been vaccinated either with a viral vector vaccine or with a nucleic acid vaccine. Most of us probably have received an RNA vaccine, but RNA vaccines are not the only types of nucleic acid vaccines. There are also DNA vaccines. And if time permits, I'm also going to tell you a little bit more about DNA vaccines that are used both in human medicine and also in veterinary medicine. So the blue arrows are pointing to vaccines that most of us have received so far. The green arrows point to vaccines that some of us may or may not have received, and the red arrows point to the type of vaccines that I would imagine many of us have already received over the last less than a year or so, since December of 2020. So generally speaking, in terms of what we are looking for in vaccines, vaccines should be safe. Obviously, safety is first and foremost. They have to be protective. There is some level of threshold that they have to confer protection. They have to be able to give sustained protection. So we want a vaccine to last a long time, perhaps several months, hopefully several years. And there are some vaccines that confer protection up to several years. But unfortunately, there are some vaccines that are very short lasting, and they can only last for, their immunity could only last for maybe a few months or so. We'll see what happens with COVID vaccines, but if anything, I would probably put my money on the possibility that COVID vaccines are not going to be long-lasting vaccines, so they have to be boosted every so often. They should be able to produce different types of immune responses. And by now, because of the COVID pandemic, we've all heard about neutralizing antibodies. We've all heard about T cells. The reality is that most vaccines produce I would say, a balanced set of neutralizing antibodies and T cells. Sometimes they produce some arms of immune response better, and sometimes they induce other types of response or arms of immune response less. But generally speaking, we should be able to strike a balance between antibodies and T cells. In terms of other considerations, there have to be very low cost, especially for veterinary vaccines. We're talking about a few cents or probably even fraction of a cent for each dose of a vaccine. Obviously, this is quite different in case of humans. And there has to be a vaccine that can be easily administered and also easily deployed. And I'm going to come back to this later. Despite all the work that we've done since Louis Pasteur in 1885, or perhaps even going back to the Chinese attempt for vaccination in 900 AD, the reality is that there are a lot of diseases for which we have not been able to make any vaccines or make any effective vaccines. There are some vaccines available, for example, against tuberculosis that is listed here, but that vaccine has actually lost its efficacy. So at the moment, I don't really think that we have highly efficacious vaccines. There are also other diseases that are not just that are not caused by just one virus or one bacteria, such as, for example, respiratory infections. And global mortality of respiratory infections is around 3 million individuals a year. The reality is that we do have some vaccines, but not necessarily very robust and efficacious vaccines against the plethora of pathogens that can cause respiratory infections. So there is lots of work to be done. And I hope that by now we've all come to realization that things can be done in terms of making better vaccines and also accelerating the process for approval of vaccines through regulatory bodies. This is just immunology 101. If I were to give this lecture let's say 18 months ago, probably I would have had to spend tons of time talking about different types of immune response and how they're generated. But by now, I think everyone knows quite a lot about the immune system and immune responses. But generally speaking, the immune system is composed of two arms that work in a very concerted manner together. The so-called innate responses that are generated or induced within a matter of few minutes upon exposure to antigens or to vaccines or to pathogens. And there's also the adaptive immune responses that are generated within hours to a few days or sometimes a few weeks subsequent to exposure to a pathogen or subsequent to exposure to an antigen derived from a vaccine. The reality is that in terms of vaccine responses, we are very specifically focusing on adaptive immune responses, and adaptive immune responses are generally speaking mediated by T cells and B cells. B cells are the ones that produce antibodies, and T cells are the ones that help B cells in order to produce those antibodies. They also can do other things, such as, for example, they can kill infected cells if they are exposed to an antigen. If they see the antigen, they learn how to respond, and next time around, when they see the same antigen, they can kill the cell that harbors that antigen. So you can imagine that if you have a vaccine that stimulates T cells, then the T cell becomes activated, and then upon activation, it can kill a cell that could become infected with a virus at a later time. This is just a very schematic representation of what goes on. There's a vaccine that is being injected. The vaccine is just full of a variety of different types of goodies for the immune system, be it a viral vector like AstraZeneca vaccine, or be it like the Pfizer vaccine that has mRNA in it, or like any other conventional vaccine, either killed or attenuated vaccines. Those are basically picked up by a type of cells that in immunology we call them dendritic cells. Dendritic cells are these cells which have projections. They're you're looking at them in purple. They pick up the antigen, they process the antigen, and then they decorate their cell surfaces with the antigen derived from the vaccine. And then they present those antigens to T cells. T cells become activated. And as I said before, they help B cells to become activated. B cells undergo massive proliferation, and then they produce tons of antibodies. And these antibodies are the ones that are absolutely critical for mediation of immunity against a pathogen, be it a virus or a bacteria. And by the same token, T cells can also produce killer T cells, and those killer T cells can kill virus-infected cells. So my next slide is about routes of vaccine delivery. So the slide that I just showed you indicated that there is going to be an injection into the muscle. But the reality is that there are a wide variety of different methods for vaccine delivery, such as, for example, the intramuscular or intradermal. There are also mucosal deliveries of vaccines, modes of vaccine delivery, such as oral, nasal, rectal, vaginal, or ocular, meaning that you can put it into the eye, especially in chickens. The reality is that most vaccines for humans are either intramuscular or intradermal. We don't really have a whole lot of mucosal vaccine delivery systems. Whereas in veterinary medicine, we've actually come a long way. We do have mucosal vaccines, and some of them are commonly used in veterinary medicine. Some of them have actually been used for decades. Like I said, ocular vaccines have been used for decades. Spray vaccines, aerosol vaccines, they've been used for decades. So we are actually, in a sense, I would say, far advanced compared to human vaccinology. But in addition to some of these ways of vaccination, there is also fetal immunization in mammals, meaning that you can essentially immunize the fetus, not used all that much. Whereas in chickens, you can easily use the in ovo route, in ovo route for immunization. As a matter of fact, many countries have adopted that, and the poultry industry has really embraced the in ovo immunization route on the day day 18 or 17 of embryonation, especially for Marek's disease, and more recently for a combination of Marek's disease and other types of vaccines, including ILT, including ND, Newcastle disease, or IBD, infectious bursal disease. So there are a multitude of vaccines that can be done now in ovo. But in addition to all of those, there is also another delivery method that would have been really nice if we could use those in the context of COVID-19 pandemic. These are epidermal or transcutaneous delivery systems. And I'll try to explain some of those in the next few slides. So this was something that I should have perhaps talked about maybe just a second ago or so. This is an OvoJect machine, probably an older machine. There are now more sophisticated machines, but nonetheless, the technology is more or less the same, which, as I said before, is to immunize the chick embryo around day 18 of embryonation, and the injection goes into the amniotic fluid, and then the chick would swallow, or it would be exposed to the antigen, and then eventually the antigen would find its way through the chick embryo's body. And by the time the chick is hatched, it's partially immunized in the sense that the immune system has already kicked in, the innate immune system has already kicked in, and some small components of the adaptive immune system have also become activated. So there is going to be a much accelerated form of induction of an immune response towards some of the pathogens that the newly hatched chick would have to deal with as soon as it arrives in the bar. So that's the in ovo vaccination route. The second one that I just wanted to touch upon is delivery of vaccines via the skin. And there are probably, I would say, more than a dozen different ways of doing this. Jet injection, which is the top panel, fourth one from the left, is actually something that is now being used in humans. So I just became aware that there are DNA vaccines that are being used or will be used as of next month, as of October, in children in India. And these DNA vaccines are going to be injected via jet injection. So this is not a science fiction anymore. Probably a few months ago, I would have said, there's a lot of work to be done, but I can tell you that now people are focusing on these sorts of vaccines and vaccine deliveries for humans. And I think the same sort of principles can be used for animals, either livestock or for poultry. So having said all of that, what are the research gaps? And I think, again, and I keep going back to the COVID-19 situation because it has really uncovered a lot of issues that we've had with vaccines and vaccination. And I think this was obviously a disaster for humanity, but at the same time, it has really truly accelerated the research process and innovation process for vaccination. So the question that we've always asked in immunology, can we expect 100% protection from vaccines? The answer is absolutely not, because most vaccines can be highly protective, but reaching 100% protection is almost an impossibility. Can vaccines protect against both infection and disease? And this is something that has come to light because of, again, the pandemic and SARS-CoV-2. The answer is most of the time, no. Vast majority of vaccines are actually made against disease and not necessarily against infection. So that's why we need to be cognizant of that and perhaps work on vaccines that would prevent both or would reduce both infection and disease. Can vaccines prevent transmission of microbes? In most cases, I would say to some extent, not completely, not fully, but hopefully they can reduce. And I would say maybe vaccines of our future would have to be able to substantially reduce transmission of pathogens. Could they provide protection against a family of microbes or different strains or variants of a virus? I think that's a wishful thinking, but it's possible. And there is some work being done to determine whether or not this can be done. For example, in case of influenza viruses and having a universal vaccine against all types of influenza vaccines. We need to make vaccines cheaper and easier to use, but can we do it? And I think if there is a will, there is going to be a means to do it. And if we can go to my next slide, I'm going to tell you a little bit more about what can be done. My apologies if this slide is not showing well here in slideshow, but suffice it to say that it essentially tells you that in case of veterinary vaccines, we've been really leaders and pioneers. We've done something that in human medicine, nobody has actually been able to do. And by the way, that picture of a pig down there, it's just a Yes, exactly, Charlotte. This one, it's not just for pigs. It's actually both for pigs and also for poultry. There are a lot of vaccines using novel platforms, such as, for example, vector-based vaccines and also DNA-based vaccines or subunit vaccines. As I said, we are probably a few years ahead of human medicine in terms of the types of vaccines that we can produce in veterinary medicine and use them commercially. Generally speaking, these vaccines are cheaper and they're easier to use. So I would argue that probably we've done something that now human medicine specialists would have to come and understand and learn how they've been done and how we've been able to make commercial vaccines at a fraction of a cost of a more conventional vaccine that can actually be used very effectively against a plethora of different pathogens. So this is, in my mind, a major breakthrough for what we've done in case of veterinary vaccines. But we are not quite done yet. Are we ready for emerging livestock diseases? And I can tell you we have a lot of livestock diseases that are emerging or re-emerging. And I would hazard a guess here, but I think in the next few years or so, we are going to be seeing influenza viruses coming back. Hopefully, they're not going to be back with a vengeance, but you never know. Viruses have their own mindset, and I always use the example of viruses versus immunologists. Who's smarter? I would say hands down. Viruses are much smarter than an immunologist or a virologist. It looks as if they have both read our immunology textbooks, and as a matter of fact, they must have written our immunology textbooks because they know everything that we don't know about our immune system. I can tell you that there are two types of viruses, family of viruses, that would be on the top of the agenda for causing some form of pandemics, either in humans or in animals: influenza viruses and coronaviruses. But these are not the only types of viruses. We also have African swine fever that, thank God, is not in Canada, but it's possible that it could be coming to Canada. Are we ready for it? And I can tell you that we are not quite ready for influenza viruses, coronaviruses, African swine fever, foot and mouth disease virus, not quite ready for it either. And we have other diseases that sometimes they reemerge, such as BSE, TB, etc. So what can we do? Again, as I said, now we have this luxury of looking at what is really in the realm of possibility. Now we know that in a matter of less than a year or so, we can not only develop a vaccine for humans, for human medicine, we can also commercialize it, we can get it through all the regulatory processes, and you can produce billions of doses. So that's easy to say, but not so easy to do for veterinary vaccines. Why am I saying it's not easy to do? I'm just going to take the example of influenza viruses. Influenza viruses have killed millions. Just for a point of reference, the pandemic in 1918 probably killed around 50 to 100 million people. And most of those individuals were in their 20s, 30s, 40s. And on top of that, the other issue with influenza viruses, unlike this coronavirus that we are dealing with at the moment, influenza viruses can jump from one species to another. So they can really wreak havoc in the process. And as a result of that, it's very difficult to control them. Thankfully, we have not had any recent influenza pandemic with a virus that's jumping from animals to humans and vice versa, going from humans to animals. But if it happens, I think it would be a disaster. And as a result of that, we need to do something about it. But what can we do? What we need to do to fight emerging diseases, we need to have cost-effective vaccines, less than a penny. If it's more than a penny, it's not going to be affordable. We need to have novel platforms for antigen discovery, whether it's big data, machine learning, artificial intelligence, what have you. We have to have those. We need to have better and more effective methods of administration and site-specific delivery, such as, for example, the use of needle-less systems for vaccine delivery. We need to have better adjuvants. We have a handful of adjuvants that are being used these days. We need to really expand our horizons and use potent adjuvants that are safe and very efficacious. We need to have vaccines that can reduce transmission of viruses. Generally speaking, vaccines can reduce transmission, but they cannot completely obliterate transmission of viruses. And we need to start thinking about what can be done to reduce both transmission and completely obliterate it. Next one, please. We also need to have thermostable vaccines, or vaccines that can withstand heat. As you can imagine, if vaccines are to be deployed to remote areas of tropical or subtropical destinations, they have to withstand heat. We have to be able to rapidly develop them and approve them. We also need to have a very robust system for deployment of vaccines. And we need to have infrastructures across the globe for manufacturing and distribution. I'm not going to go into any sort of details of politics of vaccines, but can you remember all the vaccine nationalism not too long ago? That's actually, that's been quite problematic. And last but not least, we need to have a better system for data collection and analysis, and how to integrate all of these into a robust system for a decision support system that can look after all of these things and be able to come up with strategies for control of future pandemics and emerging livestock diseases. That's all I have to say. Thank you very much for your attention.
Thank you very much for that. We do have a number of questions for you that we'll start it right off. That if you could create a vaccine for any livestock disease, what would you choose based on the impact to animals, industry, and human health? I think you somewhat answered that, but
this is actually hitting close to home for me. I would say hands down influenza viruses. Our next pandemic is going to be an influenza pandemic. Hopefully it's not going to be a pandemic with huge amount of mortality in livestock or in humans. But if I were to look into my crystal ball and make a prediction, it's going to be an influenza, and it's so difficult to make good vaccines for influenza. But we need to come together and make better, more universal vaccines for influenza.
well, and actually that leads right into one of the questions as to what type of vaccine would you use, would you make for influenza, keeping up with the changes, and is there a better strategy?
So I think, RNA vaccines are becoming a thing of the present and perhaps thing of the future. I would say RNA vaccines would probably be one of the best platforms possible for a number of reasons, one of which is the ease of making these RNA vaccines. You can actually make them within a matter of 2 days or 3 days. So you don't necessarily have to isolate the virus, inactivate the virus, et cetera, which would take a significant amount of time. So turnaround time is fantastic. And on top of that, in the past, we would have had to deal with all the regulatory burdens and hurdles. And the reality is that all of those hurdles are basically resolved now. So we can essentially jump in and create those vaccines using an RNA platform and just get them through the process, through regulatory processes.
Okay, another one. So in looking at the industry, there's some diseases, take foot and mouth, for example, that we have a vaccine for, but we don't want to use it because it's hard to differentiate between the vaccine and the exposed animals, and trade could be curtailed. So are there any techniques coming that would allow determination that you've been exposed by a vaccination versus actual exposure to the pathogen?
Yeah, so Jean, very good question. Yes, indeed. And there's a technique called DIVA, and there are actually some DIVA vaccines available that can be used, not necessarily against foot and mouth, but generally speaking, you can use them. And again, if one uses something like the RNA technology, or perhaps other sorts of technologies, such as, for example, a DNA technology, one could potentially create vaccines that are DIVA. The DIVA principles are incorporated into the development of those vaccines. So it's quite possible now. Using older vaccine technologies like attenuated vaccines or killed vaccines, it would have been very difficult to do that. But using more novel vaccine technologies and platforms, I would say is quite easy. But it needs, I would say, momentum within a country, and it needs, the will of a country to make those things.
Okay. Are we making any progress with developing vaccines against parasites?
Well, that's a very good question. Against some parasites, but not generally speaking. I would say parasitology and vaccines against parasites is not necessarily making a whole lot of advancements compared to vaccines against viruses, as far as I can see, but I could be wrong. It's not necessarily within my area of expertise to talk about parasitology, but there are some efficacious vaccines, like, for example, vaccines against Ineria in chickens, as an example. But against worms, it becomes a little bit more complicated because worms are obviously bigger and they're more robust in terms of how they can deal with things. So it has been a little bit, I would say, challenging to make vaccines against worms, but against, I would say, protozoa, like, for example, Ineria, it's been less challenging.
Okay. Does vaccination prevent the vaccinated from spreading the virus?
So that's a question that in immunology, whenever I teach immunology, I would say it depends. It really depends on the vaccine. It depends on the host. Generally speaking, it should, but does it actually do that? In most cases, it only reduces, it doesn't completely block transmission or shedding of the pathogen. So the answer is it depends.
All right, then. How much time is reasonably required in research with reference to side effects, especially serious side effects? I assume that's more of a human question than a livestock question, but with this topic, I'll allow it.
So if you're really talking about chickens, you're right, Gene. If you're talking about chickens and broilers, we are only talking about 5 to 6 weeks max, and then they're gone to the market. So probably side effects for the animal in case of poultry may not be all that critical, but obviously for animals like cattle and so forth, that would become an issue. For humans, obviously that's a much bigger issue. I can't really say for how long one would have to wait in order to be able to observe all various different potential side effects, because it's kind of actually like a black box sometimes. We never know until we hit some issues, and then we start investigating. I don't really think that historically we've seen vaccines that have been completely unsafe, and obviously we have not seen vaccines that are fully safe. I think there is obviously some risk associated with any sort of vaccination, but one would have to look at risks versus benefits. So I can't really answer that question with a lot of, I would say, degree of confidence. So I'm just going to have to maybe leave it at that.
Okay. So what's the prospect of successful pan vaccine, like a pan corona, for example, that would provide protection against all coronaviruses? The pig industry and PRRS would really like that, or PED would really like that.
Yeah, I totally get it. I think, in human medicine, we would also like to have it, and for chickens, because they also get coronaviruses, and new coronavirus variants emerge in poultry industry. So it would be nice if it could get a pan or universal vaccine. I would say at the moment, the prospect is slim, but I think there is hope, at least for influenza viruses, that there's been some work done, and there are some components, some molecular determinants of influenza viruses that could be amenable to creation of those universal vaccines. But we don't actually have anything at the moment that I can say it's in the pipeline and it should be commercializable within the next 4 or 5 years. But it's quite possible. It's quite possible, but the likelihood right now at the moment is rather slim, because we don't really have anything handy at the moment.
Here's a question. Do we know of any deaths in avians from coronavirus? I certainly know in the pig industry has been hit with coronaviruses over time as well, but PED being the latest one.
Yeah, so chickens, poultry in general, they become infected with infectious bronchitis virus, which is a coronavirus. And there are different types of coronavirus, different types of IBV that causes disease in chickens. So yes, they could actually die of coronaviruses, but it's a different coronavirus.
And I think I've only got one more question here. One that there's been some neat work monitoring social media and other data points to predict outbreaks of COVID. Could the same approach be taken with livestock diseases, perhaps to also indicate areas where vaccinations should or could be used?
Absolutely, 100%. And the person who was asking the question must have read my mind, because we've done some work with Dr. Rosita Dara looking at the utility of Twitter or other types of social media, and perhaps even Google, as a way of predicting emergence of influenza viruses, and also more recently emergence of COVID outbreaks. So yes, you can, and I think you should. Should it be the only tool in your toolbox for control of influenza or other types of pandemics in livestock species? No, it's only one of the many tools that you would have to put in your toolbox. I think it would come quite handy when you think about a rather robust and very comprehensive and composite modeling for detection and for prediction, and also for prevention of emerging diseases like influenza, perhaps even African swine fever, which thankfully hasn't hit us, and hopefully it won't, but I think we have to be quite ready, because neither for influenza nor for African swine fever we have any vaccines available commercially in Canada. So if the two of them hit, we have to be ready. And this is one of the ways that we can incorporate them into our strategies for prevention and for prediction.
Actually, there were some more questions here, so I'm actually gonna go a little bit over time, because I think that it's certainly an interesting topic. And it says, are modern livestock trending towards reduced immune function? If so, that having an impact on vaccine efficacy moving forward?
So that's a very good question. I would say this is probably one of these multi-million-dollar questions. There is really very little evidence to support the idea that our, let's say, poultry are getting less immune efficient, and their immune system is less robust compared to their ancestors. Some work was done in the US in early 2000, looking at chickens raised in early 2000 versus chickens that still had the same genetic makeup of the 1950s chickens. There were some differences, not a huge amount of differences. So it's not like a day and night situation. In case of other livestock species like cattle or swine, I don't really know much about those species, so I'm not going to comment on those. But in case of poultry, I don't really think that we are seeing a significant difference between modern day chickens versus, let's say, a 1950s chickens. So in terms of robustness of their immune system, probably they're on par. That's just my overall assessment.
Hey, I got another question. They start off by saying might be a silly question, and I'm going to say the only silly question is an unasked question. So we're going to ask this. But can we imagine a vaccine which could be distributed or spread by itself like a wild infectious virus?
Yes, we can imagine that, but is that actually a good idea? I think, especially with respect to attenuated viruses, they could actually spread by themselves. But attenuated viruses, by the time they jump from one animal to another animal, or perhaps from one species to another species, they could actually revert back to their virulent state. And as a result of that, is that actually a good idea to use them? I would say I would take this with a grain of salt, but it's possible. It's actually quite possible. But is that a logical thing to do, given what we are seeing, with SARS-CoV-2, the cause of COVID-19 jumping from one species to another species? Is that actually a good idea to do that?
Given the enormous progress with vaccines, is there a future role for antivirals?
Absolutely, 100%, because as I said before, vaccines are not 100%. Antivirals may be even better, more potent in some cases. I do really think vaccines, antivirals, and other things like public health measures or biosecurity in case of veterinary medicine, these are all parts of a toolbox, and that toolbox is becoming a massive toolbox. So I don't really look at them as either or. I would look at them as complementary measures.
Okay, I do have one. I'll ask one last COVID question, then the last question will be livestock. Do we know exactly how many vaccine doses we have for the people of the whole world, especially for the third world?
I wouldn't actually be able to say that off the top of my head. I know that we can produce billions of them a year, but what is the capacity of our manufacturing lines? I'm not quite sure. Do we have enough of them for the third world? I think we do, we should, but the reality is that it's highly politicized, unfortunately. So I think it's not just science, and it's partly politics and partly science. And unfortunately, sometimes politics has a bigger say compared to science in terms of vaccine distribution and deployment.
Okay, so this is going to be the last question. So how can industry build momentum for new vaccine development? That being the livestock industry.
I really want to have a chat with this person who asked that question, because I'm asking myself that same question. Cynically, I would say it needs some sort of something that would catalyze the process. And unfortunately, sometimes a pandemic would catalyze the process. Had we not had a pandemic, by now, we would have been talking about RNA vaccines and DNA vaccines or vector-based vaccines for humans as a far-fetched reality. We would have said, oh, these are too risky. Oh, forget about it. Who would actually get an RNA vaccine? But hey, look, it happened over a matter of 18 months or 18 months. Yeah, we've come a long way. So we can actually go a long way for animal vaccines if there is, like I said, I hope that it's not going to be a pandemic, but if there is a real, if we all come together and we say, hey, look, this is something that's going to happen. It's not a matter of if, it's a matter of when it's going to happen. I have absolutely no doubt that we will have an influenza pandemic, but are we ready for it? I don't really think that we are actually ready for it. So it's not just for livestock stakeholders, it's also for human medicine stakeholders that we all have to come together. And unfortunately, sometimes a pandemic would really catalyze the process. But I'm all for having a discussion about what we can do at the moment in order to really get this to the top of the agenda for our politicians, because without political will, we're not going to be able to be successful, unfortunately.
Well, maybe LRIC needs to pull together a group of people and have a discussion. Thanks, Shayan. And I quickly want to, kind of along that topic, the book I'm reading right now is called Spillover. I'm sure that quite a few people may have read it, but it was written in 1993. I'm not far enough through it, but somebody did say he predicted that the next, if it was a pandemic, it'd be a coronavirus. So, but it's certainly interesting reading. So if anybody wants, I can send out information on that. Thanks, Shayan, that really appreciate you giving up your time and the information that you provided for us today. I want to thank Charlotte Wall, who's behind back doing all the IT work for us and keeps us moving smoothly. Our next webinar is going to be on November the 9th. And it's going to be animal-free milk, meat, and eggs. And it's going to be with Mike Van Masso. So that is up and you can register for that. Thank you for everyone that stayed on. I know I went a little overtime today, but I think the time was worth it and well spent. Everybody have a great day.